All right, everyone. So I'm going to talk to you about some equine synovitis mitotherapy. And so I'll start with explaining a little bit of the concept behind what I'm going to be talking about. So a lot of our different regenerative modalities might actually be using mitochondria. The concept is that replacing dysfunctional mitochondria, which might be a component of the disease process we are treating, with exogenous healthy mitochondria would have the potential to improve both cell metabolism and reduce inflammation. And we have been focusing on how this could be used as a novel treatment for osteoarthritis. Now again, as I mentioned, we know that mesenchymal stromal cells release a lot of extracellular vesicles, and a lot of people have mentioned that that is a potential modality for their mechanism of benefit. And inside of these MSC stromal extracellular vesicles, we have seen that there is intact, healthy mitochondria. So it is possible that the mitochondria that we have seen in other studies may be being delivered by our MSCs. Now, when I got invited to this project, my collaborator, Dr. Christoph Mareks, had some in vitro data where he was looking at mitochondria transfer, so isolating mitochondria and then providing it to synovitis in an LPS-induced model, but it was in an in vitro model. And so his initial data was showing a reduction in inflammation in our synoviocytes, as well as increase in mitochondrial content in the cells. And then what you'll see kind of in the A versus the B panel, you'll see more red. Basically, the MitoTracker is staining living mitochondria red. So when I started helping out in this project, we were starting off with a safety study and then with plans to move on to an efficacy study. So our first safety study was just to kind of analyze whether or not what we were producing was inflammatory to the equine joint. As we've mentioned, many of our different regenerative medicine modalities induce a transient flare. And while those transient flares, we do see kind of they're self-limiting, it is kind of a little bit worrisome to most practitioners as well as clients. So before we started putting this product into joints, we wanted to see kind of from a safety profile, what was the response that we saw. So we took 3 horses and we injected their left intercarpal joint with autologously produced mitochondria. This is mitochondria produced from each horse's own platelets, produced in the morning and then administered later in that day. And then we're looking at the clinical and laboratory parameters over a period of 28 days. So over 28 days, we did clinical monitoring and saw no evidence of lameness at any time point. And then when we're looking at our systemic modulators of inflammation, we see kind of no significant change in plasma serum amyloid A, which is an acute inflammatory marker in horses, no significant increase in plasma fibrinogen over time. And then when we're looking at the local inflammation that was induced, we saw no swelling, no heat generated on the joint clinically, and we saw just kind of moderate increases in synovial fluid nucleated cell counts over time, as well as some mild increases in synovial fluid total protein. And we do know that serial aspiration of the joint is going to induce some inflammation. So these kind of are within the references published in the literature as kind of serial aspiration inflammation. So based on this safety study, we felt that we were not seeing any significant local or systemic inflammation with the mitochondria product that we were working with, so we felt comfortable moving forward with this modality. So we did want to take a step and kind of analyze exactly what we were isolating. So we are isolating a mitochondrial-rich fraction. We're not isolating just the mitochondria. We're basically using a commercial kit produced by Thermo Fisher to concentrate the mitochondrial fraction out of the cells. The above images, again, show the MitoTracker staining living mitochondrial red, but we also looked at some electron microscopy images, and we see kind of that canonical kidney bean shape that we associate with mitochondria, but again, are seeing other kind of cellular components in that fraction as well. Now we moved on to our mitochondria efficacy study. So we decided to use an LPS-induced synovitis model based on our in vitro data that saw benefit using an in vitro synovitis model. And so what we did is we had 6 horses that we bilaterally induced synovitis with 2 nanograms of LPS. And then we were going to inject the left joint with our vehicle control and our right joint with our mitochondria. And so at time 0, we aspirate synovial fluid, we inject with LPS, we collect blood, and we do our clinical assessment. 4 hours later, we're going to take another synovial fluid sample, and so that's going to be our baseline inflammatory time point. And then following that aspiration, we're going to inject with either our vehicle control or our mitochondria. We're going to then kind of collect all our other parameters, and then following at time 8, time 24, and time 48, we're going to collect our additional samples. And this model is fairly self-limiting, and so it is going to resolve on its own over time, and that's how the LPS synovitis model works. Now, our outcome measures are going to be our clinical parameters. However, this is kind of a minor outcome measure for us since it is a bilateral model, and the dose of LPS we were using was fairly low, and so we were not expecting to see significant lameness from it. We also were collecting peripheral blood at each of our time points and looking at the white blood cell count, as well as taking the serum and doing multiplex analysis, looking at an equine cytokine and chemokine panel. Now, our synovial fluid, we are going to do a variety of different analytes on it. So we're looking at our cytology, again, looking at our total nucleated cell count, as well as the individual cells that we see on our synovial fluid smear. And then we are going to do our multiplex assay. And then for a non-biased approach to see what we're at the microRNA, we are going to prepare a microRNA library from our synovial fluid. And then finally, we're just going to take the cells that are in the synovial fluid and look at kind of just traditional QRT-PCR. So at our initial results, we find that on our clinical parameters, as we predicted, we did not see any obvious changes noted over time. So we didn't see any significant lameness induced in either limb from our LPS injection, and we did not see kind of any significant variation in their systemic clinical parameters. So no increases in temperature, no kind of general malaise. Our peripheral blood, we looked at our total white blood count, and by and large, across the 6 horses over time, it's relatively stable. So we're not seeing any particular peaks systemically from our model. And then our 23 cytokines and chemokines, these are just a select few of different analytes that we got back. And by and large, the variation is more individual across different horses than over time. So we're kind of seeing that our model is not inducing systemic inflammation, which was actually our goal and kind of how we wanted to approach this project. We were worried that if we induced too much systemic inflammation, that we wouldn't be able to treat our individual left and right joints as independent. So then kind of moving forward with our synovial fluid outcome parameters, we have our synovial fluid nucleated cell count. And over time, we see our synovial fluid nucleated cell count is coming up. And then what we did is we actually took 5 microliters of synovial fluid and made a variety of different slides and then stained them to look and see what the cellular component was on those slides. And so we'll see similarly our neutrophils kind of coming up, peaking at time 8, and then kind of normalizing down, whereas kind of at the end at time 48, we still have somewhat of a monocytosis. So again, this is just showing that our model is working, that we are seeing induction of mild synovitis from our LPS injection. So now when we look at our outcomes from our multiplex assays, we are taking, again, 23 cytokines and chemokines and getting results back. Now, synovial fluid is quite a difficult outcome analyte to work with. It's highly viscous, and when you're working in a multiplex assay, you have all these little magnetic beads that are going to get stuck in really viscous fluid. So we actually would take our samples out of the freezer, and then we would hyaluronidase treat them immediately before putting them into the multiplex assay. So quite honestly, getting the results across these different analytes, we were quite happy that we were able to detect so many different analytes, and we only had a few wells that actually failed due to viscosity issues. At all time points, we were able to detect a variety of different analytes, and then we had some that were only detectable at kind of peak time points of inflammation. Other analytes were inconsistently detected or undetected. Now, when we look at things that we were able to detect across time, so at every time point, all 6 horses, both joints, we were able to detect that things were varying over time, but we're not seeing on these analytes a difference between our treatment and control. So we have significant variation over time, but no benefit or no difference between our vehicle control and our mitochondria treatment. And then when we look at kind of our inflammation cytokines that we think of kind of canonically with synovitis, we have our IL-1 beta, our IL-6, our TNF alpha, and our IL-8. We see that we have kind of detection of these predominantly at those peak inflammation time points, with kind of our time 8 hour being kind of the focus of inflammation, and with much of our inflammation resolving by time 24 and time 48. So we have kind of our inflammatory markers peaking at our 8 hour time point. So then just in summary from what I'm discussing is that at this point, the data that we have back, we can see that we have no systemic inflammation across this model, and we have local inflammation. Our model is working. However, based on the analytes we've looked at so far, we do not have a difference between our mitochondria and our control. Now, the analysis that is ongoing and in progress is to prepare our microRNA library, again, halted with some difficulties in our synovial processing to be able to get our microRNA library up and running, and trying to optimize that protocol, as well as doing our RT-PCR. We are also going to be able to kind of hone down based on the information we have on our 8 hour time point and kind of focus on sequencing at that time point. Now, in summary, what we're looking for, right, is we're kind of searching for a biomarker to see a beneficial response from our mitochondria treatment in this induced synovitis model. And then if we are able to identify a beneficial biomarker, then we could repeat this in animals having naturally occurring osteoarthritis or utilizing different models, and we'd be able to kind of hone down on a specific analyte to follow. Now, we do, again, comment that mitochondria might be a new regenerative medicine treatment, but it also might be mechanistically how our other modalities are actually having a benefit. And we need a lot more research to better understand what's going on. So I kind of got started in this project when they were moving into this in vivo model, but mechanistically we don't really understand fully how this mitochondria is working, what the best way of preparing it is, whether it's this commercial kit or collecting it from the extracellular vesicles from other cell types. And so in order to better optimize our treatment potential, we really need to better understand how this is working mechanistically so that we can utilize this modality. So I'd just like to thank all my collaborators and thank the horses who we, induced a very mild, rough couple of days, and then they get to go back out and live their happy lives for another few years. So thank you very much.
Thank you for a nice and comprehensive study. I just have a technical, perhaps, observation to make. I think anytime we talk about synovial fluid, one of the most significant outcome measures, in my opinion, is the rheology of the synovial fluid. I mean, that's the main function. So I mean, it's all nice and good to collect these data, very, very important, absolutely, to understand mechanistically what happens. But the primary function is mechanics, so we should study those rheological properties. You mentioned viscosity and all that, and characterize those.
Yeah.
Thanks, Jen. That was a nice talk. I just was interested in, so I don't fully understand the concept of looking for a biomarker, which I'm assuming is your microRNA, for a treatment?
Yeah, so the idea just that, like, if I was going to give it to some of the research horses in the herd, and then what am I going to use to measure that they're having a benefit other than just a subjective lameness assessment or the lameness scoring, like our objective lameness mechanisms. So the idea of just like anything that we look at, right, can be called a biomarker, right? Our RNA expression, our microRNA, our proteins, all of those are markers that we're looking at to kind of tell us in a simple way whether or not there is a benefit or a change.
Thank you very much. I'm curious to know why you chose the LPS model, and if you could elaborate on that versus, like the IL-1 beta synovitis model. I often have to decide about that.
So the reason we chose the LPS model, or the reason I felt comfortable, is that that's what we had in vitro data of the benefit of the mitochondria in. And so I guess, again, I said they had kind of gotten this project sketched out and like roughly funded, and then they asked me to run it. And when I said that, I was like, well, I want to make sure if we use these horses that there is reasonable data to support that this is a reasonable mechanism to research. And so they had in vitro data in the LPS model. They have a few other in vitro data models, but they did not translate well to the horse and were not understood in the horse.
So one quick last question. Do you know if those mitochondria, do they stay in the extracellular space, or they're being uptaken by cells?
So I think that is a big question, right? So we're giving them like a soup of mitochondria, and either the cells are going to take it up and break it down and then use it as building blocks for their mitochondria, or potentially the mitochondria could somehow get out and be used intact. That has not been looked at, and I think that that's like a very important mechanistic question is, just labeling the mitochondria that we're isolating and then seeing what happens to it when it's uptaken by the cells.